Hi, I'm Tania Lewis from Glacier Bay National Park in Alaska, and I'm going to talk to you about unraveling the mystery of the glacier bear. And I'd like to acknowledge my co-authors on a recent paper, Gretchen Roffler, Anthony Krupe, and Neil Barton of the Alaska Department of Fish and Game, and Ramona Mirage of the University of Calgary. Our study area is northern southeast Alaska, which includes Glacier Bay, which is in the orange circle on this map, as well as the town of Juneau, which is the capital of Alaska, and it is marked by the orange star. I'd like to also point out Lynn Canal, which is important in this story, which is a fjord going north-south parallel to Glacier Bay. And one of the coolest things about this area is that 18,000 years ago, it was completely covered in ice, with the exception of some of the areas on the coast, since the sea level was lower, that remained ice-free and served as glacial refugia. So there are plants and animal species that survived the ice age in these refugia, as well as ones to the north and the south, and they have since recolonized Southeast Alaska, many species, including black bears. So glacier bears are a rare color morph of the American black bear, which is endemic to North America. There are two ancestral groups of black bears that diverged over 1 million years ago and are believed to be mixing now somewhere in Southeast Alaska. And black bears are highly associated with forests. They're dependent on trees to escape from predators, including people, wolves, and brown bears. And interestingly, they are sympatric with brown bears on most of the mainland of Alaska, Southeast Alaska, but they show spatial segregation on islands, which is likely due to competition between the two species. Two subspecies of black bears have been identified, but still need to be sorted out, and it's not sure how color morph may play into subspecies designation. So black bears generally, over most of their range, are largely black colored, but they can also be cinnamon or brown colored, especially in the arid parts of Western United States. And in Western British Columbia is a very rare color phase of a white black bear known as the Kermode or spirit bear. And then up in northern Southeast Alaska, we have an even more rare color morph called a glacier or even blue bears. And these bears can be gray to white to black with silver tips or even blue, and mostly observed in northern Southeast Alaska and a little bit in northwestern British Columbia and southern Yukon. So what do we know about glacier bears? Well, it turns out, until recently, not very much. Our project began in the early 2000s in Juneau, Alaska, where we had a pretty big bear problem, but it wasn't really a bear problem. As usual, it was a people problem in terms of trash, and a lot of bears were getting into trash, and some of these bears were glacier bears. There were up to 6 nuisance glacier bears relocated, and several glacier bears were even hit by cars during this time frame. And Neil Barton, who is the area biologist for the Alaska Department of Fish and Game, began collecting samples for genetic analysis, knowing that there was something pretty special about these bears. Meanwhile, in Glacier Bay National Park, I started digging into the park archives to write the park's first bear management plan, and I inherited a map of glacier bear sightings and harvest locations from 1952 to 95. And this map was compiled by Linda Wiggins, who had conducted visits to the region to attempt to determine the range of this rare color morph. About 10 years ago, at the 3rd International Human Bear Conflicts Workshop in Canmore, BC, Neil, Linda, and I met with others over dinner to discuss glacier bears, and we decided that we really needed to focus some effort on learning more about these mysterious creatures, like how many are there? Are they a unique genetic population or subspecies even? And what is the conservation risk to this rare color morph? So the first step was to digitize and update the sightings and harvests. And this formed the first compilation of the geographic range of glacier bears, which basically extends from Yakutat Bay in the upper left part of this slide down to Juneau, the lower right, with a few observations in BC Tatshenshini-Elsek Park. One thing I want to point out here is that in the northern part of Glacier Bay, we don't have trees yet because it's so recently deglaciated, and so there are no black bears there. But otherwise, black bears range throughout this general area with the exception of the ice fields, of course. So I began digging into any other information I could find and putting together some clues. And we know that black-colored moms can have glacier offspring and vice versa. So it's likely a recessive genetic mutation that accounts for the color phase, and that's what's been found in the Kermode or spirit bear. One of the first people I talked to was George Ramos, a Tlingit elder from Yakutat. And he said that traditionally glacier bears were considered a third kind of bear that were smaller than both black and brown bears and possibly mated with black bears. The Tlingit name for these bears was Siknoon, which translates into a bear that disappears in reference to their small size, their elusiveness, and their ability to blend in with snowfields. Due to their rarity, Ramos claimed that glacier bears were not hunted as were black and brown bears, and elders advised that they look at them and leave them go. So glacier bears were not hunted until outside trophy hunters came to the area in the 1940s. 1909, Wilfred Hudson Osgood from the Chicago Field Museum of Natural History described a handful of glacier bear skins and skull fragments collected from Lynn Canal to Cape St. Elias, and he found glacier bear skulls were smaller but otherwise similar to other black bears in cranial characteristics. Another account from Joe Eibach, a miner in Reed Inlet in Glacier Bay in the 20s, 30s, 50s. He apparently shot two glacier bears around 1930 and described them as quite different in appearance and behavior from other black bears. He said they were smaller, rangier, more secretive, and tended to favor high barren country. So to investigate potential size differences of glacier bears, I looked at Alaska Department of Fish and Game harvest records from 1960 to 2005 in northern Southeast Alaska and found no significant skull size differences between glacier pelage bears and other black bears. So that confirmed earlier findings that there was no size difference between the color morph. Glacier bears are highly prized trophy species in Alaska. There's about 1 to 4 glacier bears harvested near Yakutat every year and about 1 every other year in the Juneau area, which accounts for 0.4% of the black bears harvested in Southeast Alaska. Sometimes glacier bears are killed in life, defense, and property as well in the cities, especially Juneau and Skagway. But it's important to recognize that glacier bears are protected from legal harvest in a large portion of their range encompassed by Glacier Bay National Park, as well as Tatshenshini-Elsek Park and Kluane. So our basic questions were, what populations of black bears do glacier bears belong to, and how are these populations related? And what is the relationship between genetic population structure, landscape, and hair color? To begin to answer these questions, for about 12 years, park staff and Fish and Game staff collected DNA samples from harvested bears outside of the parks and hair samples within the parks. The green outline here denotes Glacier Bay National Park, and to the north of that is Tatshenshini-Elsek and then Kluane. So those are the area where hair samples were all collected, and then outside was a combination of harvest and hair samples through various studies that were happening. So what we got was 114 hair samples and 170 tissue samples from black bears across the entire range. And on this map, this shows our samples locations by hair color. The purple dots are unknown. The black dots are black-colored bears. The brown dots are brown-colored black bears. And then the blue triangles are glacier bears. In the population genetic structure results, we found 10 populations largely divided by either marine fjords or ice fields. And interestingly, the populations that contain glacier bears, which included up by Yakutat, Western Glacier Bay, North Lynn Canal, and then Eastern Lynn Canal, including Juneau. Those are the only 4 populations that had glacier bears. And it was interesting that they did not connect geographically. In fact, this whole area in the middle, known as the Chilkat Peninsula, had no glacier bears. And that was true for the sightings records we had earlier, as well as the genetic samples that we collected. Interestingly, the Chilkat Peninsula is the least glaciated area in the region. There are no large ice fields adjoining this area, as opposed to the Yakutat, West Glacier Bay, Northern Lynn Canal, and Juneau area are all but 2 large ice fields. These results suggest either an unsampled population or perhaps a selective advantage to proximity to ice fields. So what is the relationship of glacier bears and glaciers besides the name? Do glacier bears survive at higher rates in ice-covered areas because their gray coats act as a camouflage to protect them from predators? If so, such an association would increase the conservation risk of the color morph as glaciers recede. And also as glaciers recede and forest cover grows and black bear ranges expand, different populations will be coming into contact with each other, and glacier bear genes could effectively be diluted. So we still have lots of questions. What is the genetic basis for the color? What proportion of glacier genes are present in each population? And where did these animals evolve? What's the relationship with ice ages and glacier refugia? And how can we focus protection to preserve glacier bears as glaciation in the region decreases with climate change? We're working with some collaborators from Hudson Alpha Institute for Biotechnology and the University of Memphis to hopefully determine the genetic basis for the glacier color morph. It looks like it's not the same as Kermode, so it's a little more complicated. And then we'd like to look at the frequency of glacier bear genes in different black bear populations in our study area. Thank you very much. I'd like to also thank the National Park Service for funding this project and Alaska Department of Fish and Game for collecting samples and for their expertise in the analysis of the data, and all staff members who helped collect samples over the years, and all the photographers who took these amazing pictures that I got to show in my slideshow. Most of all, I'd like to thank the bears for tolerating us. And at this point, I'll take questions if you have any. Thank you.
Protected areas have long been the primary tool for wildlife conservation, but only 15% of the planet is protected, meaning a lot of wildlife occur outside of protected areas. Global carnivore populations are in decline, and contact with humans is a major source of adult mortality for many species. Retaliatory killing due to real or perceived threats can cause private lands to act as mortality sinks or further isolate populations. However, private lands could also connect protected areas, provide buffer zones around them, and support wildlife. It is for all of these reasons that this Andean bear research focused on private lands here in Ecuador. Just to give some context to where we are, here on the map, you can see Ecuador is in the northwestern corner of South America. Ecuador is a small country about the size of France. It is one of the most biodiverse countries in the world, but also one of the most densely populated in South America, with one of the highest rates of deforestation. One of our most iconic species here in Ecuador is the Andean bear. It is the only ursid in South America. It is a taxonomically unique species. It has a distinct short muzzle and small ears. They are opportunistic omnivores feeding on bromeliads, bamboo, palms, and other plant species, along with fruits, insects, and vertebrates. They have also been known to eat carrion and kill livestock. They use a variety of habitats from montane forest to paramo, the high altitude grasslands, but perhaps this species is best known for its ornate or spectacled patterning around its eyes, muzzle, and chest, where it also gets the name spectacled bear or oso de anteojos. These photos do a good job of displaying this patterning that is unique to each individual bear. This is truly an Andean species. Their distribution matches that of the Andean mountain range from Venezuela down through Colombia, Ecuador, Peru, and possibly northern Argentina. They typically occur at altitudes from about 1,000 meters to 4,000 meters above sea level, although there are some exceptions. And similar to other tropical bear species, Andean bears are globally at risk, listed by the IUCN as vulnerable, with a small and decreasing population of around 2,500 to 10,000 mature individuals. They are also a CITES Appendix 1 species. In Ecuador specifically, we can see how their estimated distribution matches that of the western and eastern mountain ranges, but is separated by human activities in the Inter-Andean Valley, including the capital city of Quito. It is estimated that only a little over 50% of the bear population occurs within Ecuador's protected area system. And this estimate in itself is an argument for how important it will be to include conservation on private lands in the development of conservation plans. Threats to bears in Ecuador match global threats to the species, but specifically habitat loss and fragmentation is driven by the expanding agricultural frontier and the expansion of the mining industry here in Ecuador. Human-caused mortality is a concern. Bears are occasionally hunted for local use or byproducts are sold on the illegal market, but most significant is retaliatory killing after crop raiding or real or perceived attacks on livestock. With this setting in mind, my co-authors and I developed a research project on Andean bear presence and conservation on private lands in the southern sector of Sangay National Park. Sangay is one of Ecuador's largest parks and is recognized as an important area for bear conservation. In 1992, the southern sector of the park was extended over top of privately titled lands. And as these private lands overlapped by park boundaries, that was our study area. Our project objective was to determine if private lands with relatively low levels of human-bear conflict and suitable habitat for bears could complement Andean bear populations in protected areas, or if proximity to humans was a deterrent and private lands along park borders further isolated bear populations. We asked the question, how do human influence and habitat features affect bear presence? So this is kind of a quick photo summary of our study area, and our work would have been impossible without the support and assistance of Fundación Cordillera Tropical, a local NGO that's very active in this region. The people in this area included Mestizo and indigenous Quechua groups. Landowners in the area included individuals, families, and cooperative groups. There were domestic dogs, livestock, which was mostly cows and sheep, and one large alpaca ranch. In addition, there were large infrastructure projects in the area and a hydroelectric dam project, and this government-sanctioned road that was built inside the park boundaries. And of course, we also had a variety of other Andean wildlife. We used camera traps to collect data on bear presence, as well as data for covariates for our models. We conducted additional surveys to collect microsite features and extracted land cover data from DEM and rapid eye images. We also collected data on landowner profiles through a very informal process and with the assistance of FCT. Here's a map of the study area minus our grid cells. The purple is the paramo, the dark green is various types of forest, light green is pasture and human use areas, and the stars are population centers with the red dots being our camera trap sites. The gray line is the park boundary, and nearly everything in this view is private land, both inside and outside of the park. So to the northwestern side of the line is inside the park, and to the southeastern side of the line is outside the park. So the first step in our study was to divide the study area into 1 square kilometer grid cells, and then we eliminated grid cells with more than 80% paramo, and we did not do any sampling of the paramo. We then went door to door to ask landowners for permission to place camera traps on their properties. And we started talking to landowners 2 months before the project started and continued for over a year. In total, we had 16 landowners of 21 give us permission, and we didn't have permission to survey 2 large properties, which resulted in a heavily camera trapped western and eastern region of the study area. The study was carried out from September 2012 to September 2014, and we have 2 years of data and over 17,000 trap days. In the end, we had 40 grid cells and we moved the camera traps around to fully survey each. We surveyed on 16 properties that joined the project through a rolling entry. And we had 124 hits from bears at 45% of our trap sites and then 65% of our grid cells. We also photo captured 14 other mammal species as well. Here's the map again with green plus signs where we observed bears. We had a fairly even distribution of bears on the landscape that we were able to survey. Again, we use models to determine the influence of human activity and landscape features on bear presence. We created covariate groups and used negative binomial models fit with integrated nested Laplace approximation or INLA. Our covariate groups were landowner characteristics, microsite features, macrosite features, land cover values, habitat quality, human influence, and physical features. We then assigned covariate groups to 4 different models using bear presence as the response variable, and we controlled for seasonal effects, slope, aspect, and study year to reduce the variability in the data, making our observational unit site season study year. We also included an offset to account for the different effort at each camera trap station. Here I include only the significant results for significant models. In the human impact model without landowner characteristics, we found that the distance to rural areas with high levels of human use had a slight signal. In both habitat models, distance to low quality habitat or secondary forest was also significant. These results are really worth noting together that low quality habitat typically occurred on the forest edge close to pastures and areas of human presence. These both support the theory that Andean bears are selecting more mature forests, which typically occur further from human activity. A 2009 Andean bear habitat study in this same area showed that bears favored habitat with trees of bigger DBH, possibly linked to the presence of epiphytic bromeliads. Also significant was distance to second and third order rivers, so rivers with 1 or more tributaries. We see a decrease in bear presence the further we get from rivers, and in our study area, rivers were very small and unlikely to block bear movement, as larger rivers do with other bear species and Andean bears as well. At other locations, Andean bears have been seen using riverbanks to travel through steep forested areas, and maybe this is what we're seeing here, but to my knowledge, this effect has never been noted or studied. Study year was our most significant result. My first thought was that we had made a big mistake, but we didn't alter our methods between years and offset our analysis accounted for camera trap failures we saw in year 2. So FCT didn't report any major changes in the study region. So we started thinking about this not as study year, but as year to year or annual changes. What we're likely seeing is that bears are moving through the study area, potentially using different resources at different times of the year. Like other bears, Andean bear movement is thought to be largely based on food availability, migrating up and down topographical gradients, and previous studies in this region show bears use montane cloud forest and páramo relatively equally. So our results indicated that the Andean bears in our study were somewhat disturbed by human activities, but habitat and access to annual changes in resources might be more important when planning for bear conservation on private lands. Private lands in our study area do appear to provide a relatively safe human-wildlife interface for bears when human-wildlife conflict is low and habitat is not degraded. Larger properties where pastures are far from habitat and that both have intact forests and access to páramo should be prioritized in conservation planning for Andean bears in Ecuador. And finally, our results indicate that the private lands in our study area could complement the bear population in Sangay National Park if human activity and deforestation of mature forests is controlled and bears have access to annual resources. Although that study concluded, this issue of Andean bears on private lands continues to be of great interest to me. In 2020, my colleague Santiago Molina, myself, and our local partners launched a new Andean bear project on private lands not far from Quito city limits. This landscape has similar characteristics to those I mentioned in Sangay, but it is already a lot more complicated with more people, more livestock, more dogs, and higher levels of conflict, along with a lot of access, heavily traveled roads, and a lot of tourism. However, it is an important landscape for conservation and connecting fragmented bear populations. We're applying some of what we learned in the South to this new study area, but we continue to learn how to conserve this species in these complicated human-dominated landscapes. Thanks for listening to my talk today. If you have any questions or comments, don't hesitate to send me an email or stick around for the moderated Q&A session.
Hello, I'm Nereyda Falconi, and today I'm going to talk to you about my research that was part of my PhD dissertation. I titled this talk, The Value of Data for Andean Bear Distribution Estimations for Peru. So I want to start this talk bringing up some questions. When we start a project, we need data to answer our questions. However, we need to think about the cost. The cost not just in money, but also in time. For example, how much does it cost to get a picture in a camera trap of an Andean bear? Although it's going to depend, it's never cheap. Then we can think about the availability of data from previous studies. However, we sometimes are not sure what data is available for our use, because the data is not published all the time. And also, not always is easy to access to this data. So in general, ecological data is not available. And although many journals make the effort to request this, it's not always guaranteed. So also large-scale ecological studies that require more data, such as distribution models, distribution estimations, are outnumbered in tropical countries by North America and Europe countries. So this is because these countries used to have agencies, organizations that gather data and make huge databases that are accessible online easily. But this is not always the case, in special for developing countries such as Peru, that doesn't have this kind of data sets or organizations that are doing this work. So I realized that in order to use data or reuse data, this data need to be available. And I start a data compilation. I started contacting 59 people, 40 researchers, and also directors of protected areas in Peru. I make a research and literature review in Google. I also look in gray literature, in books, in dissertations, and in environmental reports of the ministry in Peru, and I was able to compile certain data. And with this initial data, I make some preliminary models, and I start identifying areas where the model show potential occurrence of Andean bears, but they were lacking of records. So I used this area to select my study areas to collect new field data, and also install camera traps in those areas. So this approach allowed me to focus in field gaps based in the already compiled record. After compile already collected data and collect my own data, I create a open database for the Andean bear in the Global Biodiversity Information Facility. I did it with help of my university, and I include detailed information of the origin of this data, the collector, and now it's available to be downloaded and use it just by creating an account in this platform. After this, I wanted to update the Andean bear distribution in Peru. So those are the previous studies that estimate the Andean bear distribution, and I saw the need to make an update. Those use different methods, but I wanted to make updated estimation, but also I wanted to know if this new data and published data was improving the estimation of the Andean bear. So for this, I use distribution models, and I use 3 data sets. I use one data set with only published records. I use another data set with published and unpublished records, and a third one that was also published and unpublished record, but was controlled by sample size. So they have the same number of records than the database that only had published records. I use environmental data. After some exploration, I use land covers from the vegetation cover map of the Ministry of Environment, and I use elevations. So I run models with GLM, with random forest, and with Maxent, and also I made some ensembles models for each data set. Later, I validate these models with threshold independent and dependent statistics, as I use kappa and specificity and sensibility, and also compare the estimated areas. So when I run the models with GLM with the only published data, you can see in the first picture that the estimated area was slightly small than the others ones that had unpublished data. The difference were concentrated in the center area. The other thing is that when I use the unpublished data, the areas look more connected. And well, we had a good AUC, so the model was fit well. With random forest, we saw the same pattern. Like the first model that was only with published data show smaller and less connected areas than the others. The same with Maxwell, although it's less different than the previous one. The previous one showed a bigger difference. And finally, the sample models that are basically the mean of these three first models. So it shows that when we use unpublished data, there is a larger estimated area besides to be more connected. Thus, unpublished data seems to reduce the sample's bias, which result in this more continuous, bigger predicted area. For better visualization, I create these models that are the sample models, and I just make the difference between the areas when only use published data and when use published and unpublished data. So the first one is the difference between only published and unpublished, and the second one is only published and unpublished, but controlled by sample size. So as you can see, it's the same pattern. You have a larger area in green, I'm sorry, in blue, and is concentrated in the center area. So finally, I'm showing the model, ensemble model with published and unpublished data, and I'm comparing with the estimated area by the IUCN. Also, I included the records that I use, so you can see that there are areas that wasn't included by the IUCN that is included by the models, and also there are records that are outside the IUCN estimation. Additionally, you can see that the published data are more gathered than unpublished data because the sources of unpublished data came from different sources. Meanwhile, the published data used to came from projects that have more time or protected areas that have more projects there. So this is all. I don't know if you have questions, and thank you for listening today.
Hi, my name is Emily Puckett. Thanks for clicking on my talk. I'll start by giving an overview of microsatellites as a genetic marker, then show data from our study where we surveyed allelic richness and heterozygosity from 284 populations of bears. Our data highlights spatial patterns of genetic diversity within species, but also overall levels of diversity among species. Further, by synthesizing data across studies, our results show understudied geographic locations and the species where genetic diversity data is limited. Microsatellites are a region of DNA characterized by simple repeating motifs. These motifs may be from 1 to 4 base pairs long, and due to slippage during DNA replication, will expand or contract at a locus over time. So in this example, this strand of DNA has 4 repeating units, and due to slippage, the other strand has 6 repeating units. And so this individual would be a heterozygote in the genetic data. Biologists have taken advantage of this type of DNA to turn it into a marker, specifically useful for identifying unique individuals within a population and estimating genetic diversity of populations. Bear biologists were early adopters of this technology to study the relationships among species and within lineages. Use expanded to understand population diversity, and now we see heavy usage in mark recapture studies to estimate effective population size and density of animals on the landscape. Our study identified 266 loci have been developed for bears, of which 66 of those are exclusively for the giant panda. In our paper, we argue that there are plenty of markers, and new efforts should focus on genotyping populations instead of marker development. Although I do see a caveat for Andean bears, which I'll discuss later. Microsatellites come with unique challenges that prevent direct comparison of study results. One way to get around these challenges is to genotype samples from another study to calibrate the results. And this has been used on a small scale when researchers know they want to compare studies a priori. But I'd like to argue that when you zoom all the way out to the widest spatial scale, that you can use per population estimates of allelic richness and heterozygosity to identify broad scale patterns of genetic diversity. I am not arguing that studies can be combined to infer population structure or detailed evolutionary patterns, but at range-wide scales, genetic diversity patterns are apparent. In our upcoming study in Ursus, my colleague and I do just that. What we did specifically is surveyed 104 papers that used microsatellites and extracted summary statistics from these papers. We stopped data collection in mid-2019, therefore studies published since that time have not been included. The data that we collected included sample size, number of loci genotyped, and then allelic richness and heterozygosity in each population. If we could get this information on a per locus basis, we did that. And if not, we used the estimate averaged across all of the loci in the study. We then applied a yardstick transformation, which is a technique that is meant to compare levels of genetic diversity between two populations when they use the same loci, even if they haven't used a calibrating data set. The yardstick transformation broadly showed that the patterns observed in the raw data were consistent in the transform data. So most of the results that I'll be showing today are the raw data and not the yardstick transformation. But please see the paper for more details. When thinking about genetic diversity in populations, we know that allelic richness declines faster than heterozygosity. Thus, when considering the genetic management of populations, it may be helpful to compare these two metrics to assess if a population has lost genetic diversity. Here, I've plotted allelic richness on the x-axis and heterozygosity on the y-axis, and each panel represents a different species. The points represent populations, and the regression lines are only really to aid the eye to see trends in the data. This figure gives us a strong idea about how these metrics are related to each other in bears. And I think the American black bear data is particularly useful because the species has been so well studied. And what I see is that heterozygosity essentially asymptotes within the data, such that both populations with high allelic richness and moderate allelic richness could have heterozygosity levels that are about equal. I'd like to show data for a few select species, and first let me orient you to these figures. Panel A shows average allelic richness across all loci used per population and is consistently scaled 1 through 12 across the figures, where low genetic diversity is shown in yellow and high in purple. Panel B is expected heterozygosity, and it is scaled 0.25 to 0.83 across all of the figures, again with yellow high and purple low diversity. The extant ranges are shown as either gray or yellow polygons on the maps. Giant pandas have a limited distribution with 3 subpopulations across 6 mountain ranges in central China. There are no strong spatial patterns in this data, as allelic richness is fairly even among subpopulations, although heterozygosity does show that it's elevated in the Western populations compared to the Eastern populations. Sloth bears are one of the least studied species, as only a few tiger reserves within the central portion of the range have been investigated. Within this region, both allelic richness and heterozygosity are moderately high. For many species, mitochondrial diversity gives us an indication of the number of within species lineages. However, that data does not exist for sloth bears. Thus, substantial work could be done to delimit and conserve genetic diversity across this species. American black bears have the most data, and we can observe two interesting spatial patterns. First, there is low diversity in southern populations, even though these are two different within species lineages. So we see low diversity in the eastern and western portions of the range. I would argue that these patterns are driven by high habitat fragmentation and the legacy of carnivore persecution. By contrast, we see higher diversity in the north, and that is particularly true for the heterozygosity metric. Beyond the continuous habitat and large population sizes supported, I would argue that admixture between the eastern and western lineages has increased diversity of microsatellite loci. I'm of course combining my knowledge of black bear genomics with this microsatellite diversity data set, but the species highlights how having this type of range-wide data can increase the inference that of how we understand spatial genetic diversity and therefore inform conservation and management. I want to end with looking at the Andean bear because I think it's maybe the most impactful results from our paper, particularly in terms of future conservation and management. Andean bears had the lowest allelic richness across all populations and tended lower than other species in heterozygosity. Early microsat studies with Andean bears suggested that the loci developed for Ursus were biased in this significantly diverged species. Our data supports the idea of ascertainment bias in loci used to study Andean bears. Specifically, the yardstick transformation normalizes to a reference population such that if diversity were equal to that reference, then a population would sit at 100% the level of diversity of the reference. The Andean populations are low in the raw observed data, as well as the data once they are yardstick transformed. Thus, even though I mentioned earlier that continuing to develop loci when the community already has substantial resources developed may not be the best use of conservation monies, I don't think that that's true for Andean bears. Fortunately, a reference genome was published earlier this year, and this should facilitate locus development into the future. I didn't have time today to detail all of the species nor highlight some of the subtle results from our paper. So please do check that out when it comes out in Ursus. I'd like to thank the Helen Hardin Honors College at the University of Memphis for supporting my undergraduate co-author, Isis Davis. Thank you for listening to my talk. Please feel free to reach out with questions.